Hi students, today we are going to see the types of leukocytes and its functions. So let us start with the competency. So this is the competency which is going to be addressed, which is Physiology Competency 2.6. Describe the formation of WBC, structure and function of various WBC types and their regulatory mechanisms. So today we are going to see the following objectives. So at the end of class, you should be able to list the different types of WBCs. You should be able to differentiate leukocytes based on its morphology. You should be able to list the functions of leukocyte, and you should be able to enumerate the causes for leukocytosis and leukocytopenia. So to begin with, we all know our bodies are exposed continually to the bacterial, viral, fungal and parasitic infections. So in order to combat these infections, our bodies has developed a defense mechanism. So our bodies have a special system for combating the different infectious and toxic agents, and this system is composed of blood leukocytes, which is also called as the WBCs or white blood cells, and the tissue cells which are derived from the leukocytes. So these cells work together in two ways to prevent the disease. So one is actually by destroying the invading bacteria or virus by phagocytosis, and the other method is by forming antibodies and sensitized lymphocytes that may destroy or inactivate the invader. So coming to leukocytes per se, so they are mobile units of the body's protective system, and they are formed partially in the bone marrow and partially in the lymph tissue. And they are transported to the blood to the different parts of the body where they are needed. Coming to the types of leukocytes, based on the presence or absence of granules, leukocytes are broadly divided into granulocytes and agranulocytes. So coming to granulocytes, granules will be present in the cytoplasm and it can be stained. And all the granules will have nucleus which is lobed, and they are biologically active substances which are involved in inflammatory and allergic reaction. The various types of granulocytes are neutrophils, eosinophils and the basophils. Now coming to agranulocytes, as the name suggests, it lacks visible cytoplasmic granules and the nucleus is single lobed. And the types of agranulocytes are lymphocytes and monocytes. So this is how the various leukocytes appear in the peripheral smear. So you are able to make out the RBCs in background. So always compare the size of the leukocytes with that of RBCs. So now let us see how to identify each and every leukocyte. So if you can remember this particular table, you will be able to identify the various cells under a microscope. So if you see neutrophils, they are double the size of RBCs. The normal size of RBC is around 7 microns and neutrophil is around 10 to 14 microns. And the nucleus is lobulated. Maximum 5 lobes will be present. And if you look into the cytoplasm, you'll be able to make out fine violet to pink color granules. And the distribution of neutrophil is around 60 to 70%. That means if you count 100 cells, around 60 to 70 cells will be neutrophils. Coming to eosinophil, again eosinophil is double the size of RBC and most of the times it is bilobed. So whenever you look into eosinophil, look for a spectacle shaped nucleus which is connected by thick strands of chromatin. So coming to the chromatin strands, it will be thin or narrow in case of neutrophil and it will be thick in eosinophil. So that is one of the differentiating point between neutrophil and eosinophil. And another important differentiating point is the presence of coarse brick red to orange granules. And the frequency of eosinophil is comparatively low. It will be around 2 to 8%. The next granulocyte is basophil. So again basophil will be double the size of RBCs. Here you will not be able to make out the nucleus properly because it will be masked by the granules. And you will be able to make out very coarse deep purple granules in case of basophil. and the percentage of basophil is the least, which is about less than 1%. Then coming to agranulocytes. So agranulocytes are the lymphocytes and monocytes. So lymphocytes will be of two types. One is small lymphocyte and the other is large lymphocyte based on the size. So small lymphocyte will be same as that of RBC size. So it will have a single round nucleus which will almost fill the cell, and you'll be able to make out only thin crescent shape like light blue cytoplasm, and there will not be any visible granules. Coming to large lymphocytes, again it will be more than double the size of RBC, and you will be able to make out a single round nucleus which will be central or eccentric. Again 80-20 ratio of nucleus cytoplasm will be seen. That is 80% will be nucleus and 20% will be cytoplasm. Coming to monocyte, it is the largest cell among the WBCs, more than 2.5 times that of an RBC, and the nucleus is horse shaped and it will be indented, and you will be able to make out abundant muddy blue cytoplasm, and there are no visible granules. The percentage of small lymphocyte and large lymphocyte put together is around 20-30%, and monocyte is around 8%. So if you can remember the size, the nature of nucleus, the nature of cytoplasm, and the presence or absence of granules, and the differentiating features of the granules, you will be able to identify the leukocyte under the microscope. Now coming to the functions, neutrophils are the first line defense mechanism. So they are the first responders against bacterial infection. And eosinophil will be raised in parasitic infections and allergic reactions. And basophils, they will be increased again in allergic and antigen response. The basophils are the leukocytes which will release histamine causing vasodilatation. And lymphocytes, it includes B and T lymphocytes, and you all know B lymphocytes will mediate the humoral immunity and T lymphocytes will mediate the cell mediated immunity. Monocytes respond to chronic infections. So actually the monocytes are not much active in the circulation. So they reach the tissues, get themselves modified into tissue macrophages, which will attack the microorganisms. Now coming to leukocyte levels in the blood, the normal levels of leukocytes, that is the total WBC count, is around 4,000 to 11,000 cells per millimeter cube. So there is no difference between the male and female values as far as WBC is concerned. But you all know RBC count varies with the gender. Coming to the abnormal leukocyte levels. So increase in leukocytes is called as leukocytosis, and decrease in leukocyte count is called as leukopenia. So leukocytosis generally indicates infection, and leukopenia is abnormally low leukocyte level and commonly caused by certain drugs. Coming to the causes for leukocytosis. So physiological leukocytosis occurs due to diurnal variation and immediately after muscular exercise, after meals, emotions, labor, menstruation, and adrenaline injection. Also, it will be more in the newborn and high altitude. Pathological leukocytosis occurs in all types of fever except viral and typhoid fevers. Leukemia, I'll come to that little later. Starvation, metabolic disorders like diabetes, uremia, pyogenic infections, myocardial infarction, and any tissue necrosis. Pathological leukopenia occurs in aplastic anemia, tuberculosis, viral infection, exposure to radiation, typhoid, antimetabolites, and megaloblastic anemia. So as I said, leukemias are malignant diseases in which the presence of immature WBCs in the peripheral blood occurs. And the increase in WBC count will be more than 50,000 per millimeter cube. And these are the types of leukemia like AML or acute myeloblastic leukemia, acute lymphoblastic leukemia, chronic myeloid leukemia, and chronic lymphoid leukemia. So we have come to the end of this particular class. To summarize, leukocytes are classified into granulocytes and agranulocytes. Neutrophils are highly phagocytic cells and are the first line of defense against acute pyogenic infection. Eosinophils and basophils defend the body against allergy. Monocytes are phagocytic and defend the body against chronic infections. Lymphocytes participate in immunity, and leukemia is the malignancy of leukocyte precursor cells. So just to check whether you have understood or not, kindly answer the following questions. So name the leukocyte which acts as a first line of defense against infections. Yes, correct. The answer is neutrophil. The next question is, name the leukocyte that has bilobe spectacle shaped nuclei with coarse orange-red granules. So kindly note the coarse orange-red granules. Yes, very good. It is eosinophils. Name the leukocyte that is largest in size with kidney-shaped eccentric nuclei with abundant bluish cytoplasm. So here the catchy word is largest in size. Yes, correct. It is monocyte. The last one, try to identify the leukocyte. Are you able to make out the size of WBC? It is double the size, and you are able to make out many lobes which are connected by thin chromatin filaments. Okay, so it is a, yes, correct, neutrophil. Thank you.